Hello and welcome. In this episode, Dr. Jonathan Golub and I are continuing our conversation on genetic screening tests for cancer. For interested listeners, I'd recommend listening to our earlier podcast. There we discussed the differences between diagnostic and screening genetic tests, targeted and non-targeted tests, as well as direct-to-consumer tests versus tests that were prescribed by a physician. There was a lot going on, so do take a listen. We also discussed what you're likely to see in a test results. And there we discussed what a variant was and the distinction between pathogenic, benign, and unknown variants. In this episode, John and I will be discussing how an expert in genetics or bioinformatics goes about investigating a test results. So what we're going to be doing is to actually take a genetic test result and see how a geneticist or a doctor would actually explore this test result. We hope you'll find this useful. Hi, John, and welcome again, and thanks for taking the time today.
Hello, thank you for having me. I've been looking forward to this.
Awesome. So how do we get going? So let us start with discussing what one would see in a genetic screening test report. Can you help us? And if you want to do a screen share, John, just go for it. So
wonderful, yeah. And what you get back as almost the raw data, raw analysis out of a genetic screening test is a list of suspected or identified changes in the genome of that patient that was analyzed versus what is found in a reference genome, or the sort of standard off-the-shelf human genome. And there are several of them. They're up to version 38 in the most recent.
And the reference genome, correct?
Yeah, exactly.
Yeah.
Starting from when more or less I started in science in the late 90s, there's been successive generations that are with varying degrees of completeness and representation of the mixture of human genetics. But either way, what you end up with is sort of a list and then a shorthand that is used to describe when there is a variant. And there's more ways to have variation than we originally talked about in the first podcast. In the first podcast, we focused on a fairly narrow but important kind of variation, which is when there's a change in the part of a gene that is involved in encoding that gene's amino acids. And that DNA sequence has this idea of degeneracy. So there's 3-letter codons that each correspond to an amino acid, but each amino acid can have most of the amino acids can have more than one codon all going to the same place. So there's some tolerance of even mutation in there without a change in the protein coding sequence. And you can see that, and that's one of the shorthands you'll get. You'll have this amino acid was this, but in this and the reference genome, but in this patient, it's this amino acid instead. And that's sort of your most concrete, direct way that you can have a variant. It is now the amino acids, the thing that make up that protein, are different. You can also describe this in the level of DNA. The DNA at this location is no longer in an A, it is a T.
Yes.
And that may or may not have an effect on the protein coding sequences because of that codon word thing having multiple codons mapping to the same amino acids. But that's another way to describe it. And in fact, you can describe the same variant in either way. And then even adding additional complexity within the realm of DNA, you can have this notion of splicing. And so you can have the gene as it is after it's converted to RNA and then further spliced down to the messenger RNA that then goes off to be converted into protein. And you can have addressing, the base pair of RNA at this location is different from the reference genome.
So or you can have to investigate all of these, right? So when you get a test result, you're going to have to look at it with respect to the reference genome. You're then going to have to really, dig into how this variant is described, whether it's at a splice site or not, and so on and so forth. I'm excited. We're going to be digging into these things today.
Exactly. And so maybe a good example here is to look at one of these shorthands that would come back from a patient.
Yeah,
and talk about it, and then we'll dig into it.
absolutely. So what shall we start off with? Shall we look at a pathogenic variant, or shall we look at a variant of unknown significance? You choose.
Maybe let's go ahead and do a pathogenic variant, maybe
Mhm.
of a gene that's known well, and we'll go
Mhm.
for that one.
That sounds good. So here is one that you and I have been discussing for a bit. Do you want to share your screen, Jonathan?
Sure. And let's go look at it. So here we're looking at a gene in the BRCA
Shoot.
2
Yeah.
um, gene. And you can see here again, the naming scheme is a little bit silly. This is a
Mhm.
website that is put out by the National Library of Medicine, which is a service of the National Institutes of Health, and in turn, the Health and Human Services in the U.S. federal government. This is one of the core repositories. And you can see here we can search and have a whole bunch of information. And this is all of the different repositories that are of information that are maintained
John,
by.
before we dig into this, so if you could just scroll back to the top.
Mhm.
So I just want to kind of emphasize that what we're seeing there, what you're highlighting there, is actual results from a genetic test report, correct? This is what, a patient potentially received when they had a genetic screening test done, correct? And now we're going to the next step, which is we're taking that verbatim from the test report and investigating it.
Exactly. And
Mhm.
so here you can see in the BRCA2 gene,
Mhm.
there's a deletion.
Deletion, yeah.
And here, because it's a deletion, we're going to do it within the realm of cDNA. And
Mhm.
it's deletion, and you can see the numbers here. And that's starting from the start of that gene's DNA, cDNA.
Mhm.
And again, cDNA and all this terminology comes a little bit from the realm of, is this post splicing or not, and everything else.
Mhm.
And so it's a deletion, which is another way. It's not a change in a base pair, but missing DNA relative to the reference genome. And it's been seen a lot of times. There's many, many papers that have worked around this gene or its variation. There's a whole bunch of sequences, actual gene sequences and human genomes that have been deposited as a part of hundreds of studies showing this. But really, when you kind of come into it, this ClinVar is a separate thing that often my eyes start to turn to, because this gets into the variance and how it is understood. So again, BRCA2, you can see the DEL, and you can see again in the protein space, it's a missing lysine. They've done a little bit of translation with the two of the more recent reference human genomes. It's on chromosome 13 and a whole bunch of numbers into it. There's no protein change, right? Because it's a deletion. These last three columns are where things I think get to what a patient would really want to know. What cancers is it, or conditions is this variant been associated with? So in this case, breast ovarian cancer and familial susceptibility. It is a pathogenic variant, and it has been reviewed by an expert panel. This is about as firm as one can get for a variant. It is solidly demonstrated to be um, pathogenically related to increased risk for breast cancer and ovarian cancer, even after review by an expert panel, and been observed many, many, many times. There are certainly lots of tools, and tools more oriented when you encounter a new variant that really hasn't been encountered or hasn't been studied or described. But this is a nice resource to be able to get into and see what it looks like. And again, this is a variant. It isn't like a change in the amino acid. This is a deletion, but decidedly one that has been seen many, many times and very much clinically associated with a risk of cancer. And if we
John,
go into Mhm.
sorry, I didn't mean to.
No, no, go
I think
ahead.
I think I think our listeners would be interested in, understanding more what we see in the first column of this table. You spoke to the reference genome a little while ago on number 37 and number 38. Maybe we can just point out how it's different, and what constitutes a reference genome and why it's different.
Yeah. So what the reference genome is in
Mhm,
essence the DNA sequence of the whole human genome, all of the chromosomes in the human genome, and then often a little bit of some of the accessory DNA. So very common viruses that are found in human cells and everything else.
mhm,
And they start at the beginning of each chromosome and go all the way through the middle and then out to the other end of the tail. And I think people might remember that human chromosomes and human DNA, unlike, say, bacterial DNA, is not circular. They're linear. So they have a start, middle, and an end. And in the middle is often where, after the DNA is copied before a cell is divided, it matches together. So the ends of human chromosomes in the middle, so the ends of the chromosomes in the middle or centromere of a chromosome are almost more not a site where typically things like protein coding genes are located, but instead they're structural parts of the chromosome to help with things like cell division and copying chromosomes when cells divide. And as such, a lot of the DNA there, the sequence of DNA there is repetitive. The telomeres are even made by an enzyme called telomerase. So it's a repeated sequence over and over and over and over and over again. So through these successive generations of trying to sequence human DNA, the number of repeats and the variance in those repeats has changed a little bit. So if you're going to try to describe something as being the, you know, 329th, sorry, I think this is like 32 millionth base pair.
mhm.
The telomeres, the end of that, the chromosome is going to change a little bit with each version as we get better and better at really characterizing them. Likewise, the middle of the chromosome, or centromere, that also tends to have a lot of repetitive sequences that are more around helping the DNA match up with itself as it does cells undergo replication, or things like sperm or egg cells are made, and events like homologous recombination occur when the genes are mixed up with one another. Those repetitive sequences are just difficult to sequence. You see the numbers here, where this BRCA gene is located and where this variant is located. It's a little bit different between version 37 and
37
38.
and 38, yes.
And it's as we've gotten better at, like, no, there's not that many variants of that, this many variants. In contrast, the, lacking a better way of putting it, but the meat of the chromosome, not the centromere, not the telomere, but the part that actually contains almost all of the protein coding genes and non-protein coding genes that are involved in human physiology, those tended to have been a little bit more stable over time because they're easier to sequence, easier to assemble and put together. And that means that that's how they then are able to kind of shift things around to get it. And so this is almost like a placeholder. If you can download, anyone can download from the genome website put together by the National Institutes of Health, you can grab the whole of your version 37 or 38 genome and grab the 13th chromosome within it and zip over there. And in fact, if I recall how this works, that is exactly what one can do if you wanted to go ahead and do that, is actually start to look at it. You can see here genomic location, and then there's multiple versions of each build of the genome. UCSC also puts out a version of it, but you can go and actually see it in the context of the chromosome. So chromosome 13, and then you have a sort of address, point 1. Here's all the genes that are there, and then all of the variants that have been there, and you can see all the way through here. And so when we think about the human genome, there's a further notion that there isn't a human genome, but all the genetic variation of all the people who exist. And
Mhm.
so there's an attempt here to combine all that together and make a consensus, but then recognize what are normal, healthy variants and then abnormal variants. And so you can see here, some of these involve copy number variation, some of them involve all of it. And this is just a way for one to be able to go through and see this in context. Conversely, if one goes back here, you can do the same thing, but now in the older version, this is version 37. We're still in chromosome 13. We're still 13.1. But you can see here there are subtle differences between it, including the variants that went in to do it. And so, and this gets a little bit back to how a lab would identify variants. And so this would be like whole genome sequencing and then aligning back to a reference genome versus more targeted sequencing of protein coding sequences or exome sequences or whatever else. And so that was what
I loved, I think, just a little bit more on the screen and then we can continue. But this is a 4 base pair deletion, and you and I were discussing how, this deletion results in a change in the amino acid lysine and why that's interesting. Maybe we could talk a little bit about that too.
Yeah, and this is even more than just merely affecting lysine. Lysine is an amino acid that can be modified post-translationally, which is important. But the words of DNA that go into making an amino acid, the building blocks of a protein, they're 3-letter words. So if you're missing 4, what this causes after that point in the gene is what's called a frame shift mutation.
Yeah.
Because all of the words are different there. So this results in a massive change in the gene, at least at the protein coding level. And so if the gene is important, that gene is very likely to not function, at least in the same way, if not be completely unmakeable at all by the cell. And so that's what's going on here with the 4-base pair deletion as compared to a 1-base pair or 3-base pair deletion, which would remove an amino acid but leave the rest of the protein intact. And even that, it depends on the phase where those 3 base pairs were deleted. And that's what's going on here. And so coming back to it, this is a kind of frame shift mutation that is just so absolute in a gene BRCA2 that is very important in suppressing breast cancer. And so that's why, this is one of the best studied genetic susceptibilities to cancer. And so we kind of know a lot more of the study. And even here, you can see it's not all the way to 4 stars, which is fascinating in and of itself. Because again, if you're going to tell someone that they have cancer, you really, really, really want to be very, very cautious about saying something. So the difference between the 3-star is that this has been reviewed by an expert panel versus 4, where it has been integrated as part of practice guidelines, such as a change in screening or prospective mastectomy in this case. And this particular variant has not reached that level of certitude. It is clearly pathogenic, but has not yet had the sort of studies done to support it being driving by default a change in care. That does not eliminate a good discussion with someone's provider about doing something about it. And that's
I think that's a very interesting point. So you have a patient who's received this in their test report. It's not yet a practice guideline, so it's a matter of what the patient's risk tolerance in determining what the next steps are. I mean, of course, it's a collaboration with the physician, but there are no practice guidelines that say, hey, you've got this BRCA2 mutation, get a double mastectomy or an oophorectomy, right? There's not a practice guideline that gives you this information crisply, and so that puts the patient in kind of a state where they have to kind of decide what their risk tolerance is and communicate this to their oncologist, correct?
Exactly. And getting back to even some of the things we had touched on in our first podcast together, if the motivation for ordering the test was, say, a family history, a close family member who had breast cancer, either aggressive or discovered early or both, and then one orders a genetic test and this result comes back, it all is telling a very same story. And that's where having a discussion with a trusted physician to really interpret it, and then add in the person who is tested, age, their risks, their risk tolerance, and their desires. But this is information that's very, very congruent. This very much speaks in the same way to that family history, and would, I think, help clarify a bit more about subsequent decisions about what to do for that person. and would be frankly a quite concerning pattern to have that mutation with it. The other thing I point back, again, speaking back to our original thing, as I said, this is one of the better understood cancer susceptibility genes. The assessment of this variant was last done in 2016. And so this is why having that raw information, the sort of address, this is a deletion, 4 base pair deletion in the BRCA2 gene. Going back to one of these sites and re-looking at the interpretation of it matters. The degree of pathogenicity, it can, with further study, can be better clarified. Things can go up in that star. So it stars as just a variant that was observed but of unknown significance. Then it becomes something that's likely pathogenic, then quite compellingly pathogenic, or increasing the risk for, in this case, breast cancer. And then so compellingly that it has become part of practice guidelines. If someone is identified as having this mutation, they should strongly consider increased screening and or prospective mastectomy.
Yeah, and
when
that is a nuance we didn't discuss last time, which was so obvious with this particular variant, correct? That even if it's pathogenic, doesn't necessarily mean that it's percolated into the realm of being a change in what you would do with that result. So I think that's an important nuance there, and unfortunately another level of uncertainty for patients who might receive this in their genetic test reports. So so one other thing you and I were discussing a little earlier was we discussed how in this particular mutation we're dealing with lysine. We looked at why that was interesting. So in your experience, how often do you actually look at this, these variants and say, hey, I want to see whether it has any effect on the protein structure? I mean, I know that this was a 4 base pair mutation. Maybe we can go through one example where there's a actual change in the protein structure, which can be problematic, or maybe not. So,
yeah, maybe I'll give another example, and this is on a building on to that, where we are looking at here we are. This is another variant we can see, and again, I'll put into the direction here what we're looking at. This is a mutation in the you can see it up here where it is. It's C7502A to G, but in the gene ATM. So ATM gene, you can see.
Sorry to interrupt you, Jonathan, but again, this is also a result that we are taking from an actual patient's genetic test report. So this patient had a genetic test for breast cancer, and they received this report that said that they had a mutation in the ATM gene, and we're feeding that into the same website, correct?
Precisely. And you can see here that this particular variant was first submitted in 2016. It is last evaluated in August of last year, 2023. And different groups that have submitted it, so it's been seen in more than one study. People have disagreements about what it means, but this is a little bit more of what we were talking about. It's a single nucleotide variant. It's only 1 base pair long, and it is a missense. So in other words, this does affect the protein, the amino acid in this protein. The protein will still be made, but you're going from an ASN or asparagine to serine. And so, and you can see all of the papers that have done it. This was, and you can see people had different arguments about what disease they were even considering. So there's some broadly cancer diagnosis, others it's sort of other degenerative diseases, and maybe they don't mention it. But overall, the general consensus is that some people are saying, it's probably benign. Some are making no arguments about it. But either way, this at least has been observed before and has popped up in it. And so there's a nice way to kind of come over. This is a nice summary of it. About these different conditions and this particular change in amino acid. So why am I bringing this up? In comparison to and comparing it, in comparison to the BRCA2 variant that we looked at before, which truncated the protein, had a massive change, and then further has been clinically associated with increased risk of breast cancer. This is a variant, it's a single base pair variant. It will change the protein, just one amino acid in the protein, however, so it's a less drastic change. It's been observed multiple times in multiple studies, which is another thing we bring up. There can always be artifacts, right? Maybe the sequencing machine for that patient's specimen missed something or made an error. And so you just want to be sure that this is a variant. And when the variant that is observed in that particular patient has been observed multiple times in the past, that does make us think a little bit more that at least it is real. And then its significance is very much up in the air here.
Yeah, and it's interesting for us to discuss this because, this is actually from a patient without a family history of breast cancer who's going through treatment for breast cancer. So could we go back to the previous tab, please, on the conditions?
Oh, yeah, of course.
Yeah. So, I would love for our listeners to, take note of the third column here, which is the number of submissions. So you're a practicing physician, you take care of cancer patients. How much can I trust this? How often is this information updated? I think that's a question we get, don't we? So maybe we can speak to that, because I don't see, that there's familial cancer of the breast right here, I think, which is the 6th row. If you scroll down, I don't know if there's more here. There is none. So there is a single submitter, and the number of submissions is 1. And when you look at the last evaluated column, I guess the point I'm trying to make is this is not consistently updated, is it?
It's consistently updated in the sense that the National Institutes of Health does have some automated tools. So if a study uploads a bunch of human genetic data from study participants, it will look and see are these variants still existing. And of course, there's lots of different studies. So 3 of the submissions, 3 different projects that all had sequence data that showed this mutation, they didn't even specify what they were looking for. Like, why did they do the genomic sequencing, right? Whereas this study, they were looking for, these studies were said, hey, we're looking for cancer predisposition syndromes, or likewise a familial history of cancer of the breast. And so you are right? It's the other half of it. It's not the sequence data, but the information about what was known about these. Did this person have breast cancer? Did they have a family history of breast cancer? Did they have later on go on to get breast cancer? And so that ends up being a sort of a tricky question. And you can see here I'm clicking into this to kind of go into it. So this variant exists. It is a change in the protein that everyone agrees exists, and it's been observed many, many times. 2 different groups have submitted it, and they've done it as part of clinical testing. One has sort of said, it's likely benign. The other one made no statement about it. And at the end of the day, that is incumbent upon someone else to go and do the full study to try to understand it. And that means someone would have to go, and there's a couple different ways. So how to phrase it? There's much cheaper, faster ways to screen a large number of people for a specific variant than sequencing the entire genome. So let's say you gave a pot of money And you did that. One way to do that is to do a case control style study where you take people with a family history of breast cancer and people without, and look to see is there enrichment for this particular variant in the people with a family history of breast cancer. One could do it as a cohort study. One could follow people a long time. So there's a whole bunch of studies, but someone needs to do that study to interpret it. So coming back to an individual patient, someone has this condition, has this variant, and and maybe they had the testing done because of family history of breast cancer. If I saw these results, this is a variant that is observed. It's been considered. Maybe some groups think it's benign. Maybe some groups are just not sure about it. I would not be particularly reassured by this discovery, and I would probably, if the discussion with the patient was already increased surveillance or increased concern based on the family history, nothing in this test result would change me from that thinking. And that's why I would go ahead and, based on what we have available to us, I would continue to go. I would not view this as reassuring, which is in contrast to that BRCA2 mutation that we started with, where I feel like then in some ways it's not good news, but it is at least congruent information, in the sense that then I would be feel like the counseling for the patient would be that there is a risk, and we have at least one evidence for genetic risk down at the sequence level that fits with the family history. Whereas this one, the family history is inconsistent with this, but this is not a reassuring finding. The fact that it's likely benign or uncertain significance would not make me feel particularly reassured for this particular patient.
Okay. I think
Yeah.
a couple of other things I would love for us to just talk about. Population frequency analysis. I don't think we quite dug into that in this conversation about both of these variants. So let's just discuss that, because I think it's interesting to kind of just consider.
Yeah, and so there's this notion, there's a bunch of different terms for it and lots of different ways of doing this, but I think this concept makes a sense sort of intuitively. So if you have a big room of people, some of whom have increased risk and some of whom do not have increased risk for breast cancer in this case, if there's a gene or a variant of a gene that is associated with breast cancer risk, what you would expect is that it would segregate with breast cancer. So if the overall prevalence, 1% of the people have this variant in the whole room, you would expect that the people who will not go on to have breast cancer, who do not have breast cancer, would have less than that. And the people who do have breast cancer or are destined to have breast cancer will be much more enriched for it, maybe 10 times higher. And a little bit of how enriched that is, it has to do with how risky that particular variant is and how it interacts with other genes and all of these other complicating factors. But at the end of the day, there's a couple different ways of doing this. Like I said, one can look for the prevalence of a variant in cases, people with breast cancer, and controls matched as well as you can without. Or you can do it as a cohort. You take the room and then divide that room in half, where the room is selected, and by definition, you don't have to do any matching. But for very rare conditions, that is the other style, where you do cases and controls, is much more practical but less compelling of evidence, because you never quite know if you've managed to match people perfectly. And so an example again would be those BRCA genes. The BRCA gene was identified as being related to breast cancer by looking at families with a family history of male breast cancer.
Male breast cancer, yes.
Yeah, and where the variants in that particular gene sort of kind of pointed in that direction in those cases, compared to matched controls of similar families without male breast cancer.
Awesome. Thank you. So I think, there's been a lot that we've discussed today. We've taken a couple of variants reported on, patients' genetic screening test reports. We've done a literature search. We've looked at clinical significance, the genomic coordinates. We discussed the reference genome. We actually looked at how you would do sequence retrieval, and then the consequences of, what the mutation is. Is it a frame shift? Is it a single nucleotide deletion? And then kind of also discussed, population level frequency analyses and how that can be potentially done or not done, and the values associated with multiple ways of doing this. So anything else you and I have discussed which we've missed today?
No, I think bringing back to the focus of an individual patient, this is both the excitement and the challenge of this kind of study or these kind of tests, is that the bad news is the most interpretable, unfortunately, which is to say the variants that have clear pathogenic potential are the ones that are probably going to be the most actionable for an individual patient, whereas for all of the complexities that we've just mentioned, there's lots and lots of different ways for a gene to vary. It's pretty hard to do the study, to accumulate enough patients to understand the significance, particularly of rarer variants, that I would expect the default answer for most variants that come out of a genetic test to be unknown significance. And even those that are likely benign, that can change over time. And so if there's any utility is to go to this, DB-SNP or related resources provided by a trusted source like the National Institutes of Health, and revisiting down the line with your physician or on your own the variants and see if their understanding has changed as more studies have occurred. Because the interpretation is very in flux, whereas the mutation is generally not. That exists or doesn't exist. It's its meanings that can change over time, and maybe in particular things that maybe seemed uncertain or benign up front can become pathogenic, and conversely things that were seen pathogenic up front sometimes can go. But looking and keeping an eye on the studies and interpretation is, I think, critical and essential.
Yeah, and today we also saw, and that's a great summary, but today we also saw one case where you have a variant that's definitely pathogenic, but has not yet, graduated to the level where there's a change in practice, which makes it even harder for the patient. So, and for the communication between the doctor and the patient. So, thanks again, John. This was a super interesting conversation, and I look forward to further conversations with you about genetics and all things to do with how we can use genetics and genomics to help our patients.
Wonderful. Thank you. I've really enjoyed it.
Thank you.